Welcome everyone. Today we are going to continue the third part of the chapter 11, the fundamentals of the nervous system and the nervous tissue. In this lecture, we are going to learn about the synapses, the components of the synapses, and its different types of transmissions. So, what is a synapse? The neurons are functionally connected by synapses, which we are also called as junctions that mediate information transfer from one neuron to another neuron or from one neuron to an effector cell. There are two types of neurons related to the synapses, the presynaptic neuron and the postsynaptic neuron. The presynaptic neuron, the neuron conducting impulses toward the synapse, is the one which sends the information. The postsynaptic neuron is the one where it receives the electrical signals. That is, the neuron transmitting electrical signal away from the synapse, the one which receives information. In the peripheral nervous system, the postsynaptic neuron may be a neuron, a muscle cell, or a gland cell. If you look at this figure, you would see the presynaptic neurons, the axons of the presynaptic neuron, and this is the receiving neuron, which is the postsynaptic neuron. That is the cell body actually. And in between, this is another neuron, another postsynaptic neuron, and in between the presynaptic neuron and the postsynaptic neuron, you would see the junction, the synapse, and overall it calls the synapse, and the space between these two is called the synaptic cleft, which we are going to learn later. The synaptic connections can be divided into majorly two parts, and additionally less common types are mentioned here. The more common ones are the axodendritic and the axosomatic. So in axodendritic, the connection happens between the axonal terminal of one neuron and dendrites of others. The axosomatic, it happens between axonal terminals of one neuron and the soma of the others. The less common connections would be axoaxonal, that is from axon to axon, dendrodendritic, dendrite to dendrite, or somatodendritic, dendrite to soma. There are two main types of synapses. They are the chemical synapse and the electrical synapse. So here it has shown the less common types. The axodendritic synapse, this is the dendrites, this is the axon, so axodendritic synapse. Axoaxonal synapse, this is an axon, this is an axon, so axoaxonal. Axosomatic, this is the axon, this is the soma, cell body of the receiving postsynaptic neuron. With related to the synapses, the chemical synapses are the most common type of the synapse. They are specialized for release and reception of chemical neurotransmitters. Typically, they are composed of two parts: the axonal terminal of the presynaptic neuron, which contains the synaptic vesicles filled with neurotransmitters, the receptor region on the postsynaptic neuron's membrane, which receives the neurotransmitters. It's usually on dendrite or cell body. The presynaptic as well as the postsynaptic neurons, they're separated by a fluid-filled space, which we called as the synaptic cleft. With related to the chemical synapses, the electrical impulse changed to chemical and across the synapse and then back into electrical. So that is how the transmission happened. The continuing the chemical synapses, the transmission across the chemical synapses across the synaptic cleft cleft is important because the synaptic cleft prevents the nerve impulses from directly passing from one neuron to the next. And this is a chemical event as opposed to the electrical event. And because of the synaptic cleft, as well as with the chemical synapses, transmission across the synaptic cleft depends on the release of the neurotransmitters, diffusion of it across the synaptic cleft, and the receptor binding of neurotransmitters. This is actually a unidirectional communication. There's no reverse happening, nothing from postsynaptic membrane stimulates the presynaptic. It's always unidirectional. So the chemical transmission across the synaptic cleft happen in several steps, and we'll go over one by one. It involves 6 steps. The first one would be the action potential arrives to the axonal terminal of the presynaptic neuron, and the action potential stimulated the voltage-gated calcium channels to open up, and because of that, the calcium going to enter into the axonal terminal of the presynaptic terminal, and this calcium entry causes the synaptic vesicles to release neurotransmitters. What is happening is these vesicles go and fused with the presynaptic membrane, and they release the neurotransmitters into the synaptic cleft. So here it shows the action potential arriving to the presynaptic terminal. The calcium voltage-gated calcium channels open up, calciums enter into the presynaptic terminal. The vesicles get stimulated. These are the neurotransmitters. They goes and bind into the presynaptic membrane and then release the neurotransmitters into the synaptic cleft. And neurotransmitters diffuses across the synaptic cleft and bind to the specific receptors on the postsynaptic membrane, and binding of these neurotransmitters to the receptors open up ion channels, and that would create a graded potential at the postsynaptic membrane. And at the end, if the neurotransmitters kept binding to the receptors, the postsynaptic membrane get stimulated, but there are ways to terminate this stimulation. It could be reuptake by astrocytes or axonal terminal, or it could be degraded by enzymes, or it could diffuse away from the synaptic cleft. So this is again the same picture, the complete picture which we have seen. So the neurotransmitters diffuses, they bind into the receptors at the postsynaptic membrane. That would lead to opening up of ion channels, create a graded potential at the postsynaptic membrane, and eventually the stimuli would terminate based on the 3 mechanisms: reuptake, degradation, or diffuse away. Talking about the synaptic transmission, there's a word called synaptic delay. Synaptic delay is the time needed for neurotransmitter to be released, diffuse across the synapse, and bind to receptors. It could be take either 0.3 milliseconds or it could be taken up to 5 milliseconds. The synaptic delay is actually a rate-limiting step of the neural transmission. And transmission of the action potential down axon can be very quick, but synapse slows transmission to postsynaptic neuron down significantly. However, this synaptic delay is not noticeable because these are still very fast neuronal transmission. The electrical synapses, it's less common than the chemical synapses. Neurons are electrically coupled. They're joined by gap junctions which connect cytoplasm of adjacent neurons. However, the communication is very rapid and it may be unidirectional or bidirectional. So that's the difference between chemical and the electrical. Electrical could be unidirectional or bidirectional, and also it's very rapid. It is found in some brain regions responsible for eye movements or hippocampus areas involved in emotion and memory, and it's most abundant in embryonic nerve tissues. So let's now talk about the postsynaptic potentials. So neurotransmitter, when it's bind to a receptor, they cause something called graded potential that vary in strength based on the amount of neurotransmitter released by the presynaptic membrane or the time neurotransmitter stays in the cleft. Also, the postsynaptic potential depends on effect of the chemical synapse. There are two types of postsynaptic potentials. We call, in short abbreviations, EPSP, which stands for excitatory postsynaptic potential, or IPSP, inhibitory postsynaptic potentials. So let's look at the excitatory synapses and EPSPs first. In this type of transmission, the neurotransmitter binding opens up the chemically gated channels that allows simultaneous flow of sodium ions and potassium ions in opposite directions. So sodium influx is greater than the potassium efflux. Efflux means going out, resulting a local net graded potential, which we call as the depolarization, and that's called excitatory postsynaptic potential. So sodium ion more into the postsynaptic membrane cause the depolarization. The EPSPs, the excitatory postsynaptic potentials, trigger the action potential in the EPSP is of threshold strength. If the EPSP, which is triggered by the action potential, is at the threshold strength, they can spread to axon hillock and trigger opening of voltage-gated channels, causing an action potential to be generated at the postsynaptic membrane. So here it is being shown in a graph. Here is the time, the membrane potential in millivolts, and this is the stimulus, and this is the threshold level. So getting in sodium causes the membrane potential to be less negative. That's why it goes towards the minus 55, and it's reaching out the threshold level. So in this case, it has not reached the action potential yet. It's closer to the action potential threshold. And with related to the inhibitory synapses, the neurotransmitter binding to the receptor opens chemically gated channels that allow entrance or exit of ions that cause hyperpolarization. So we have learned when the nerve is hyperpolarized, you have to have a very strong stimuli to make it polarized again, or it's really kind of impossible to stimulate. So it makes the postsynaptic membrane more permeable to potassium or chloride ions. And if potassium ion channels open, it moves out of the cell. If chloride ion channels open, it moves into the cell, which makes internal inside of the cell membrane becomes more negative. So more negative means it's go away from the threshold level. So the inhibitory synapses, the hyperpolarization reduces the postsynaptic neuron's ability to produce an action potential, and it moves further away the action potential. It moves further away the gradient, the electrical stimuli. You should see the electrical stimuli here. It's moved further away. The membrane potential moves further away from the threshold level of the postsynaptic membrane. threshold level here. You would see here the threshold level is the minus 55. You would receive the stimulus at minus 70. Because of the membrane become more negative, the membrane potentials goes further down. It could be maybe minus 80, minus 90, and so on. So there's also a concept called integration and modification of synaptic events. So the summation by the postsynaptic neurons. What does that mean? A single excitatory postsynaptic potential cannot induce an action potential, but the excitatory postsynaptic potentials can summate, or which means as adding together to influence a postsynaptic neuron. Like the excitatory postsynaptic potentials, the inhibitory postsynaptic potentials can combine or added together and leads to inhibition of the postsynaptic neuron membrane. So most neurons receive both excitatory and inhibitory inputs from thousands of other neurons. And only if the excitatory postsynaptic potential predominates and will able to bring to a threshold level, it will generate an action potential. So thinking of the summation, there are two types, temporal and spatial. Temporal summations means like one or more presynaptic neurons transmit impulses in rapid fire order. The first impulse produces the excitatory potential, and before it can dissipate, another excitatory potential of the synaptic potential is triggered, adding to top of the first impulse. With regard to the spatial summation, the postsynaptic neuron is stimulated by large number of terminals simultaneously, and many receptors get activated, each producing excitatory potential synaptic potentials, which can then add together. So here it will show up the neuronal integration, the excitatory potential and the inhibitory potential. So here you would see the threshold of the axon of the postsynaptic neuron, the resting membrane potential. There is no summation has happened because two stimuli is separated in time causes excitatory potentials that do not add together. Here you would see the temporal summation, where you would see before the first one weighs off, the second excitatory potential makes added into the first one, and that will go over, shoots up the threshold level and generate an action potential. The spatial summation, the two simultaneous stimuli at different locations causes the excitatory potential that add together. And remember, we discussed that's happening at the postsynaptic membrane potential. And here, of course, it shows us the spatial summations of the excitatory potential over here, and then the inhibitory potential over here. We change the membrane potential, can cancel each other out. So both happening at the same time in one neuron, as you can see it in here. So the synaptic potentiation, that's another concept that we need to know. So that is, the repeated use of synapse increase the ability of the presynaptic cell to excite postsynaptic neuron. In there, what will happen is calcium ion concentration increases in the presynaptic terminal, causing release of more neurotransmitter. And this leads to more excitatory potential synaptic potentials in the synaptic neuron. The potentiation can cause calcium ion voltage gates to open on postsynaptic neuron. with related to, it's a long-term potentiation, and it's with related to learning and memory. The presynaptic inhibition, the release of the excitatory neurotransmitter by one neuron is inhibited by another neuron via an axo-axonal synapses. You have seen the axo-axonal synapses in the presynaptic inhibition. What will happen is the release of the excitatory neurotransmitter by one neuron is inhibited by another neuron, and this is happening at the axo-axonal synapses, not with the others. So less neurotransmitters are released, and that leads to smaller excitatory potential at the synaptic membranes. The other important topic that we need to discuss with related to synapses are the neurotransmitters. Neurotransmitters are the language of the nervous system. There are 50 or more neurotransmitters have been identified with related to the nervous system, and most neurons make 2 or more neurotransmitters. Neurons can exert several influence, and these neurotransmitters can exert several influences. Usually they're released at different stimulation frequencies, and the neurotransmitters can be classified by their chemical structure or based on their function. With related to their chemical structure, we can classify them as acetylcholine neurotransmitters, biogenic amines, amino acids, peptides, gases, and lipids, or endocannabinoids. And we're going to look at one at a time. So let's look at acetylcholine first. It's the first identified and best understood neurotransmitter. It's released at neuromuscular junction, also used by many autonomic nervous system neurons and some central nervous system neurons. They synthesized from acetic acid and choline by enzyme choline acetyltransferase. So if you notice, the ones which I highlighted and underlined are very important, and they're very good multiple choice facts. And so you have to know the chemical component, the acetic acid and choline, and you can also figure that out from its name itself, acetylcholine. And it's prepared or synthesized by choline acetyltransferase, but it's degraded by the enzyme acetylcholine esterase. So don't get confused. These two enzymes, degraded by acetylcholine esterase, synthesized by acetyltransferase. The next category would be biogenic amines. That is where our catecholamines and indolamines comes. So dopamine, you might have heard these words before, dopamine, norepinephrine, and epinephrine. They are the catecholamines and made from the amino acid tyrosine. The indolamines are the serotonin and histamine. The serotonins made from amino acid tryptophan, the histamine made from amino acid histidine. All widely used in the brain, and they play a major role in emotional behavior and biological clocks, and used by some autonomic nervous system motor neurons, especially the norepinephrine. The other category would be amino acids. Amino acids make up all proteins, we learned at the very beginning. Therefore, it's difficult to prove which are neurotransmitters. But so far identified ones are the glutamate, aspartate, glycine, the GABA, gamma aminobutyric acid. The other category is the peptides, the neuropeptides, the string of amino acids that have diverse functions. They are the substance B, which mediates of pain signals, endorphins, the beta endorphin, dynorphin, enkephalins, so on. The pain perception. And the gut-brain peptides, somatostatin, cholecystokinin, they play a major role. The purines, the other category, the monomers of nucleic acid that have an effect on the both central nervous system and the peripheral nervous system. The ATP, the energy molecule, is now considered as a neurotransmitter. Adenosine is a potent inhibitor in the brain, and also these monomers can induce calcium ion influx in the astrocytes. The next category would be gases and lipids. The gastrotransmitters, that is the nitric oxide, carbon monoxide, hydrogen sulfide gases, are considered as gas transmitters. They bind with G protein coupled receptors in the brain. They are lipid soluble and are synthesized on demand. Nitric oxide involved in learning and formation of new memories, as well as brain damage in stroke patients and smooth muscle relaxation in intestines. So that's why sometimes it's been used as for anesthesia to relax the muscles. H2S acts directly on ion channels to alter function. The endocannabinoids act as same receptors as THC. Most common G protein coupler receptor in the brain, and it's lipid soluble, synthesized on demand, believed to be involved in learning and memory, and also it may be involved in neuronal development, controlling appetite, so on. So classification by the function, we can divide them into two types due to effects and the actions. And also with related to function, they exhibit a great diversity of the function. With related to the effects, it could be excitatory versus inhibitory. The neurotransmitter effects can be excitatory, we call it as depolarization, and or inhibitory, hyperpolarizing. So effects determine the receptor to which it binds. The GABA and glycine are usually inhibitory. The glutamate neurotransmitter is usually excitatory. Acetylcholine and norepinephrine bind to at least two types of receptors with opposite effects. It's important, like acetylcholine is excitatory at the neuromuscular junction in skeletal muscles, but it's inhibitory. It has an inhibitory effect in the cardiac muscle. With regard to the action, it could be direct or indirect. So direct action, neurotransmitter binds directly to and opens ion channels, which promotes rapid responses by altering membrane potential. Examples are acetylcholine and amino acids. In direct action, the neurotransmitter acts through intracellular second messengers, usually G protein pathways. It would be the broader, longer lasting effects similar to hormones. The biogenic amines, neuropeptides, and dissolved gases are considered as indirect actions. The neuromodulator, they're chemical messenger released by neurons that does not directly cause EPSPs or IPSPs, but instead they affect the strength of the synaptic transmission. And it is said that it may influence the synthesis, release, and degradation of the reuptake of neurotransmitter. They may alter sensitivity of the postsynaptic membrane to neurotransmitter, and they may be released as paracrine, and effect is only local. So now we have discussed the neurotransmitters, the type, and now it's time to discuss about the receptors. So the receptors, they are either channeled linked receptors. With related to the channel linked receptors, they are ligand gated ion channels. The action is immediate and brief. And excited receptors are channels for small cations, such as sodium ions. Sodium ion influx leads to the depolarization. And inhibitory receptors, they allow chloride ions influx that causes the hyperpolarization. Another type of receptors would be the G protein linked receptors. Their responses are indirect, complex, and they're slow and often prolonged. And they involve transmembrane protein complexes They cause widespread metabolic changes, and examples would be muscarinic acetylcholine receptors, biogenic amines, and the receptors that bind neuropeptides. And the mechanism is complex. Once the neurotransmitter binds to the G protein link receptor, activating G receptor, they activate G proteins, which is at the cytoplasm, have been bound to the receptor. This activated G protein controls the production of second messengers such as the cyclic AMP, cyclic GMP, diacylglycerol, all calcium ions, and these second messengers can then leads to either open up of closed ion channel or open or closed ion channels, activate some enzyme phosphorylation, or so on. So here they have depicted a diagram of sketch of the G protein coupled receptor. So this is the receptor, this is the G protein which is attached to the receptor. Once the ligand bind to the receptor, the conformational change happen with the receptor. G protein leads to change in its composition and leads to activation of the second messengers or formation of second messengers, and that leads to activation of several pathways. The other important concept is the neural integration. So neural integration, neurons functioning together in groups. The group contribute to a broader neuronal function. There are billions of neurons in the central nervous system, and if they worked alone, there could be confusion. So they must have an integration so that the individual parts fuse to make a smoothly operating as a whole. So organization of the neurons we call as neuronal pools. It's a functional group of neurons which integrates incoming information received from receptors or other neuronal pools, and they forward processed information to other destinations. In a simple neuron pool, single presynaptic fiber branches and synapses with several neurons in the pool, like in here, shown in this figure. In the discharge zone, the neurons closer to incoming fibers are more likely to generate impulses. But in the facilitated zone, the neurons on the periphery of the pool are farther away from incoming fiber, usually not excited to threshold unless stimulated by another source. So that's the difference between the discharge zone and the facilitated zone. With related to the processing of these signals, the serial processing happens. So in the serial processing, the input travels along one pathway to specific destination. One neuron stimulates the next one, which stimulates the next one, etc. And system works in all or none manner to produce specific anticipated response. The best example of serial processing is the spinal reflex. With related to the reflexes, the rapid automatic responses to stimuli. Reflex is a rapid automatic responses to the stimuli. It's particularly stimulus always causes same response. Occur over pathways called reflex arcs that have 5 components. It composed of the receptor, sensory neuron, central nervous system integration center, then the motor neuron, then the effector. So this is the reflex arc, starting from the receptor here, and then it brings, this is the sensory neuron. This is the sensory neuron. It brings the stimulation from the receptor to the integration center, which is at here. This is the interneuron in between the sensory neuron and the motor neuron. Once the integration happens, the motor neurons carries signals to the effector. It could be a muscle or a gland and so on. So this is a reflex arc, and this is a simple reflex arc. With related to the parallel processing, the input travels along the several pathways. Different parts of the circuitry deal simultaneously with information. One stimulus promotes the numerous responses, and this is important for high-level mental functioning. Examples would be a sense smell may remind one of an odor and may associated experiences. So this is higher order functions of the brain. The types, thinking about the types of circuits in the nervous system with related to neurotransmission, circuits are defined as the patterns of the synaptic connections in the neuronal poles. There are 4 types of circuits: diverging, converging, reverberating, and parallel after discharge. And these are the types of circuits which has been depicted in here. The diverging circuit is one input, many outputs, and amplifying circuit. It's an amplifying circuit. Examples would be a single neuron in the brain can activate 100 or more motor neurons in a spinal cord and thousands of skeletal muscle fibers. The converging circuit, it has many outputs, many inputs, one output, as it's shown in the figure. Examples would be different sensory stimuli can all elicit the same memory. The reverberating circuit, the signal travels through a chain of neurons and each feeding back to previous neuron. It's an oscillating circuit. It controls rhythmic activity. Examples: the breathing, sleeping, sleep wave cycles, the repetitive motor activities, and so on. The parallel after discharge circuit is signal stimulates neurons arranged in parallel arrays and eventually converge on a single output cell. So impulses reach output cell at different times and cause a burst of impulses called an after discharge. So examples would be exacting mental process such as mathematical calculations, so on, that parallel after discharge circuits works well. Okay, so this is the end of the presentation, and I hope that you have gained a breadth of knowledge about the fundamentals of the nervous system and the nervous tissue, and we'll meet in the next chapter. Thank you.